Hello, everyone. Welcome to the MIF++ seminar. It's a great pleasure to introduce Professor Ridgway Scott from the University of Chicago, both departments of computer science and mathematics. We will talk today about data deserts and drug design. Over to you, Ridgway, please.
Thank you very much. Data deserts and drug design. Okay, so I think drug design, you understand where I'm headed. Data deserts may not be a concept you've heard of before, but it seems like today you have to say what you're doing with respect to AI, because the assumption is that AI can solve every problem today. But in fact, we discovered that there are situations where AI doesn't have enough data. And I'm calling that a data desert. And there are different areas of science where there are data deserts. One important one is turbulence. And I find people don't understand the fact that we can't model how airplanes fly, even though they fly all the time. And so surely there must be enough data on how they fly. And in fact, that's the problem. You can't get data on flying aircraft because you'd have to trail something behind it, and that nobody would let you do that. So, but today I want to focus on data deserts in drug design and how you can remedy that by extending the protein data sets that we use all the time using simulation and physical models. Okay, so let me just start with a little bit of motivation. This is a slide from some years ago, but I remember when we first saw this, we were just shocked that somehow over a period of time, drug design was getting harder, not easier, and more expensive, not cheaper. Because typically in technology, you find that things get easier to do. It's easier to build cars with special features and cheaper and so forth. And you get used to it. Certainly with computers, computers get faster, cheaper, easier to use. But in drug design, it seemed to be going the other direction. And we said, well, why is that? What is missing here? Are there some concepts needed? First of all, what are the concepts in drug design? We couldn't figure out very many. And so we started looking at that. And we came up with a concept which we called the dehydron. So this developed over many years, and I'm just jumping in sort of at a waypoint here to show you what it looks like. So this is a typical protein backbone for two paralog kinases, meaning kinases which are very similar in shape. And there's also a drug bound in there. They're not really important at this point. But what is important is that we've marked certain hydrogen bonds. So if you don't know, you should learn that hydrogen bonds are what hold proteins together. That was not immediately obvious. It's documented in our book. It's one of the things that I'm proudest of in the book, of tracing that history of how did people come to understand that proteins really were the things that held, hydrogen bonds really were the things that held proteins together. And it turned out that hydrophobicity played a key role in that, and that could be seen in many experiments done over decades. So here are two very similar proteins, and we've marked certain hydrogen bonds called dehydrons. So some are in green for one of the from one of the proteases and for one of the kinases, and one in red for the other kinase. And what you can see, although I haven't explained exactly what these hydrogen bonds are, you can see that they're really different. Maybe not always. I mean, sometimes if there's one, there's another. But in other cases, they just don't have any match whatsoever. They're really distinct patterns. And so this tells you that these dehydrons can be used to distinguish what would otherwise be very similar proteins. Okay, and here's an example of how that was used. So there are two drugs that were patented some years ago, over 10 years ago now. And they were based on being able to separate two different kinases by two different drugs. One drug would attack one and not the other, and vice versa. And so these were the WZBZ drugs. And I mentioned this because it led to a patent. So this is just to say that we found that this whole concept of dehydron and the associated wrapping technology about it, which I will explain in a second, this did definitely have a real benefit. It really came to fruition and led to drug patents. Okay, so let's drill down a little bit more as to what we mean by dehydrons and wrapping technology. So this is the HIV protease, and it's got an inhibitor in there in the middle that's darker gray. And in blue is the backbone of the protein backbone of the protease. And marked in green are these dehydrons. So these are the ones that we've been focusing on. And then in other hydrogen bonds are just listed in gray here. So these hydrogen bonds are what hold the proteins together. They're critical, but it turns out that the green ones, the dehydrons, are quite distinctive with respect to their properties. And so that is what I want to explain in a little bit more detail. So here's that flap of the HIV protease. And what I've done is I've taken a particular dehydron, this one here, and I've drawn a sphere around this end of the hydrogen bond, that red one, and then another sphere around this end of that hydrogen bond. And what we're going to do is we're going to count in that two-sphere object how many hydrophobic objects there are, how many things that repel water. And so that turns out to be the critical determinant of what a special hydrogen bond is, whether or not the hydrogen bond is well protected from water by hydrophobic groups or other groups, but mainly hydrophobic ones, or is it underprotected so that water can come in and attack it. And just FYI, this inhibitor in here, notice it has these carbon rings. So these are very hydrophobic things, and they turn out to be right in the middle of what we call this desolvation domain, meaning they got added. And so we're going to claim that part of the binding effect of this inhibitor is that these hydrophobic groups are drawn in to protect these hydrogen bonds. Okay, so that's a lot to consume at once. Let's just again break it down.
Ridgway, could
Yes.
I ask a short question on the previous picture?
Yes.
So you said in the background with blue polygonal line is the standard backbone,
Yes.
right? And I guess the vertices are simply alpha carbon atoms.
Yes.
The nodes where two edges meet.
That's right. That's right.
And so hydrogen bond here connects alpha carbon atoms, but you mean a standard bond from oxygen atom to nitrogen, probably.
That is right. Carbonyl, yes. Amide carbonyl hydrogen bonds.
I see.
Backbone hydrogen bonds. There are other hydrogen bonds that are of interest, but right here I'm only focusing on the backbone ones, which are really the most significant.
And could you also clarify the difference between green and gray bonds?
Yes. So the gray ones turned out to be ones which are well protected by carbonaceous groups, by hydrophobic groups. So in other words, when you draw these spheres around one of these gray ones and then count how many carbonaceous groups are in there, you'll find that it's a very large number, like 30. Whereas around a dehydron, it'll be more deficient, typically 10 to 15.
So that's the key difference. So simply counting with how did you say CH groups, right?
Yes, yes. In the end, all we do is count number of carbonaceous groups inside those desolvation spheres. And I'll show you why
Mhm.
we came to that as something to do to measure hydrophobicity. That was a key point. We didn't understand when we started, what does it mean for something for the environment to be hydrophobic? And we had to figure that out. So
Mhm.
I'm trying to draw you in backwards, if you will, in this. And
Okay,
let me just keep going and show
thank
you.
you, of course.
Okay, so first of all, why is this so hard? Why am I not doing some sort of standard bioinformatics technique, just counting things? Well, because there's a lot of physical chemistry going on here. And so first of all, what you need to understand is that protein side chains have very large electrostatic gradients. They'll have a +1 or +2 charge, and it might be just right next to it as a -1 or -2 charge. And those charges are small, the order of an electron. And so you'd say, well, why is that such a big deal? Well, but they're so close to each other. So you go from plus to minus so quickly. So those are very big electrostatic gradients. Well, if it were just for that, things would be quite different. So in particular, people have done studies of protein folding in a vacuum, and you get quite a different answer. So the fact that we're folding in water, essentially, a vitreous environment, is critical. And why? Because water is a very strong dielectric. It's probably the strongest dielectric that I know of that occurs in nature. Its dielectric constant is 80 times that of the vacuum. And then finally, there are these hydrophobic groups which modify the water structure, and they're distributed in interesting ways. So let's just go through the different forces here. So we have very large electrostatic gradients, but they're screened by the dielectric effect, the effect of water. And then finally, the water effect gets modulated because of the hydrophobic effect. Hydrophobes can move water away, and so that changes the screening. Okay, so it's the balance of those three forces that we're trying to understand here. Okay, and why physical chemistry? Why, I don't know, maybe you've been to many talks on bioinformatics where people bring in physical chemistry, but certainly when I first got involved, I was a little bit surprised not to see more physical chemistry reasoning. And so I found this quote, which goes back to 1897, saying that we need to understand physical chemistry to do physiology. And so our theme is not new, but we're continuing to apply it, I would say. Okay, so now we wanted, so proteins have side chains. This is the big deal about proteins. So I don't know. There's a joke about what a nerd. What's a nerd? A nerd is somebody who has a favorite protein side chain. And if we met at a bar, I'd be happy to tell you what my favorite protein side chains are. But here are some of the most likely protein side chains to be found at an interface. So you can just examine protein interfaces throughout the PDB and see what side chains happen to be close to that interface. It turns out these are the top 7. And what's interesting is they have very little in terms of carbonaceous groups. So here is a hydrophobic carbonaceous group, and here's another one over here. But for the most part, there's no such carbonaceous group. Now, and those are the ones typically near interaction sites. Okay. Um, and here are just the opposite. Here are a bunch of the most hydrophobic groups, side chains that have all carbonaceous groups here, essentially. I mean, phenylalanine, of course, is the biggest one. And you might have thought that you would find these side chains at interfaces because people postulated there was a theory that what caused proteins to get together was the hydrophobic effect, meaning that by bringing two proteins together, you'd get rid of water, and that would make it somehow would make it likely that they would join there. Well, that turned out to be absolutely wrong. And as I'm going to explain. Now, let me say something about dielectrics. If you've never studied dielectrics, it's certainly worth study. And water is maybe the most interesting dielectric. And I argue that water is maybe the most important molecule in biology. So if you haven't really glommed onto that idea, I would be happy to talk to you more about that. But what is a dielectric? A dielectric is something that shields charges. And it's like having a fog around a lighthouse. And so, this lighthouse probably is lit up here, but we can't see it through the fog. And that's because the dielectric, the fog, keeps it from shining as brightly as it would otherwise be. Okay, so here's our situation. We've got these charged side chains. They're like lights in the distance, and they're surrounded by water, and that's the dielectric. But as can happen with the fog, things can happen to remove the fog locally. And in our case, we're looking for things which remove water. So that would be a hydrophobic group would be able to remove the fog. Okay, here's a little bit more of a quantitative explanation. We don't really have it properly quantified, but the idea is this: hydrophobic, typically CH group, CH2, 3, group 4 for methane, remove water locally. They're hydrophobic. And this causes a change in the dielectric constant locally, and that makes the potential corresponding electric potential change and makes dehydrons actually sticky. So I'll say a lot more about that. This can be quantified and used to predict binding sites. I'll say a lot about that as well. And we call the placement of hydrophobic groups near an electrostatic bond wrapping, just like you're wrapping an electrical wire that's not got any shielding on it. Okay, and you can see this effect on a single hydrogen bond. We'll try to explain that eventually. So here's the math again in more equational form. If you have this equation for the electric field and the potential, and here's the dielectric constant, and when that goes down, the solution goes up. When epsilon goes down, phi goes up, and that's the energetic benefit of being dehydrated. Okay. Now, what's a unit of hydrophobicity? We didn't know this at first. We knew that hydrophobic effects were important. And we looked around, at different parts of side chains and tried various different measures. And we came up with the simplest one and the most effective one as being a single carbonaceous group. Well, my chemistry is not great, and I wanted to understand, what does that mean? Why could it be that a single carbonaceous group could have such a big effect? Well, then I thought about things like methyl alcohol and ethyl alcohol. So one is deadly, one is drinkable. Or ethylene glycol versus propylene glycol. For those of you who have travel trailers, you put propylene glycol in it in the winter to keep the drinking water from freezing. Okay, so can we see a molecular level effect analogous to the change in dielectric permittivity? That's a question we want to answer. And what can a simple model of dielectric modulation predict? Okay, so we're going to look at that. So here is a cartoon of how we see, first of all, the hydrogen bond, the backbone hydrogen bond, and how it would behave if it were surrounded by this sphere of carbonaceous groups. So imagine the sphere is somehow packed by these carbonaceous groups and water can't get in. So water is kept out of this sphere completely. Well, so here's a couple of waters that are out there. So they would try to interact in some way, but they're they're kept at a distance from this hydrogen bond, so it remains quite strong. By contrast, suppose we lose some of those so-called wrappers, and we're left with a picture like this, where now the water molecules can come in and interact with the constituents of the hydrogen bond. And so this oxygen interacts with this hydrogen and conversely over here, and that weakens this hydrogen bond. So this we call the underwrapped hydrogen bond, or dehydron. It's something which is susceptible to being weakened by the approach of water. And so that's our concept. That's why we count these carbonaceous groups to get a rough idea of the hydrophobic environment around a hydrogen bond and any potential changes that occur with that. Now, this was an early paper that came out discussing the dehydron by someone we didn't know at all, Alfonso de Simone. And he made this wonderful picture showing simulations that he did of water around parts of a sheet prion. And he picked two hydrogen bonds, one down here that's not well wrapped, and one up here that is well wrapped. Now, it's a little bit hard to parse, but I'll try to help here. So he listed all the water molecules in the simulation, some over 9,000 of them. And he, on this axis, just for every time that a particular water molecule was near this site where the hydrogen bond is, he would put a marker there. He just put a dot. Now, if it stayed there for a very long time, as here, those dots would connect and you'd get a long line. And notice that's relatively sparse. That is, not all of the water molecules came to visit this hydrogen bond over the time of simulation. By contrast, here's one that's underwrapped, and it's everywhere dense here. I mean, it's the water molecules come and go, they don't stay very long, and so they're much different from the behavior around a well-wrapped hydrogen bond. And we had predicted this based on our understanding of the dehydron. And we were very pleased to then interact more with Alfonso. One thing I did, in fact, I said, could I get the distribution of those two hydrogen approaches that you had, the two hydrogen bonds. So the distances for the hydrogen bonds here, and he gave me this data set. And so this is the distribution of the separation of those hydrogen bonds, and as a function of the distance of that potential separation. And so there are two distinct groups. One of them is stronger. This blue one is stronger. That's the well-wrapped hydrogen bond. And then this one is the underwrapped hydrogen bond, which is looser. The distance of the hydrogen bond is larger, and it decays more rapidly. So the blue one is stronger and more stable than the red one. Okay, so all of this made good sense to us as basic physics of these kinds of hydrogen bonds. Okay, so then how do we apply that kind of thinking to to protein-protein interactions, or protein-ligand interactions more generally? So here is, let's say, an underwrapped hydrogen bond, as we've shown before, and then we bring in a ligand, somehow, magically, removing the water, and all of a sudden that hydrogen bond strengthens. Well, what happens is that hydrogen bond is gaining energy, becoming stronger, and that corresponds to a force that effectively pulls that ligand in there. So that's the idea of dehydrons supporting binding. And there are no, notice, intermolecular bonds here. This is just a indirect effect. which I'll try to explain a little bit here. So what is inter versus intramolecular? So intermolecular bonds are like the power cord on the computer. You plug it in and the electrons go across. An intramolecular hydrogen bond is something where it's more like the toothbrush charger, if you have one, where you have electrons going around here, and that sends some waves, and that induces motion of electrons here and charges your toothbrush or camera or whatever it is. Okay, so those are the two different approaches. And what we would argue is that this sort of molecular bond can be stronger than any sort of geometrically allowed intramolecular bond. So these are very natural bonds between two different, a protein and a ligand. So that's our argument for why and how we might have this kind of binding. And knowing that, then we said, well, wait a minute, if we believe that's how things can bind, then we should find examples of that in the PD, the Protein Data Bank. And so we took a look, and here's the HIV protease with an antibody. Protease here, antibody here, and notice a dehydron right here. And then when you look at the bound form of the antibody with the protease, it binds right there. It's like a finger pointing to that spot. And when we saw that, we said, well, wait a minute, maybe we're onto something here. And so we looked further in a much more complicated setting. So there are protein-protein interactions in virus assemblies, and there are many, many known. And so you can look at them and say, okay, where do these virus particles, how do they assemble their various small proteins? And this is an attempt to explain that. This is a little bit hard to parse. You have to really study it closely, but you need to know where these different virus particles come together, where these different proteins meet to form the virus shell. And invariably, there are dehydrons at these points where things get associated. And so we'll leave that to you to examine in more detail. But that was another test that we knew had to be passed, in other words, for us to really believe that dehydrons could be involved in protein interactions. And then one more test was to say, well, what is the extreme? What if a protein has almost all dehydrons, almost no strong hydrogen bonds? Well, this is the human prion, and it does have some hydrogen bonds. You can see these gray lines in here. These are well-wrapped hydrogen bonds, but then there are a whole bunch of dehydrons in green on the outside, and it's well known that they form amyloids, and believe that that's a big reason for prion disease. Okay, so one more thing about the basics of hydrogen, of hydrophobic interactions. So I asked myself, what about the code, the genetic code for coding for protein side chains? And so this is the standard way the genetic code is written. What I've done is I've added two things. First of all, I've added the number of hydrophobic groups in that side chain. So phenylalanine is the one with the most, and cysteine has none. And so notice when you do it in this way that all of these ones in this column and over here tend to be hydrophobic, and the ones over here tend to be hydrophilic. And so you know that the most likely kind of change in a mutation would be a single point mutation that would move you from one box to a neighboring box. And so it seems that the code, or it can be argued that the code, is set up to make it so that it's definitely likely that when you modify a single point mutation, you won't necessarily likely change the wrapping behavior by much. Okay, so there's a lot more detail here that I could go into, but this is a very useful way to think about things. Okay, so fine. So we got convinced that dehydrons were able to predict binding, and we used that in several papers to analyze and predict, and in patents to suggest new molecules for drugs. But then we realized we were a little bit hampered because the concept of dehydron required the full structure of the protein. And in many cases, you don't have the full structure of the protein. Well, what we realized is that there is a thing called the disorder score. There are many different disorder scores. This one happens to be Ponder. And what it does is you just take the sequence number and you feed the sequence into Ponder, and it gives you this disorder score. So up, goes down, et cetera, et cetera. So up here is disorder, and down here is order. Okay, so in this case, this protein is mostly ordered and has a disordered area. And really, let me give full credit here. It's really Ariel who realized this, that the change between order and disorder was what he calls a twilight zone. And it has to be something where you have a little bit of order. So you might have some dehydron, something not strong enough to really give it full order, but it's not disordered. So these are the spots you want to look at where it's going to change from order to disorder. And so we did that. So these are different parts of the protein that we were looking at. Here's what it looks like in ribbon form. And what we find is that, in fact, where there's these transition zones, that's exactly where you find dehydrons. Okay, so now what we can say is that we can use the disorder score not to predict exactly these unwrapped hydrogen bonds, but where they're likely to occur within the sequence. And so that tells you some sequential information. So we're saying this region here happens to be a region we believe to be changing in wrapping, changing in strength of hydrogen bonds. And so that could be an area to target in drug design. And so they did. Ariel and a colleague in Wisconsin were able, Richard Moss, were able to come up with a drug design for heart disease just based on sequence alone. Of course, it had to be tested and so forth, but the point is that that was enough information to know where to look in the sequence for possible binding spots. Okay, so that was a breakthrough. That now we felt comfortable that we could predict to some degree dehydrons based on sequence alone, at least the likelihood of finding dehydrons. Let me just mention one other thing. This is not the only thing we've thought about with dehydrons. There are issues that come up in proteins. Here's one you can look at yourself. Here are two lysines. Lysines are positively charged, and they're sitting right next to each other. And what does that mean? Why don't positive charges repel? Well, one answer might be that they're actually the charges have become deprotonated by some effect or other, and we realized that dehydrons have the ability to deprotonate, and therefore are active in enzyme catalysis. And that's a completely different subject that I'm not going to talk about, but I just want to mention it here to say, if you're at all interested in enzyme catalysis, this is a possible avenue to look at. Okay, so conclusion so far. I want to move on to data deserts. So we've been looking at data mining in the Protein Data Bank and other databases. And what it has done for us is give us this quote, a mental picture of the constitution of living matter. And so that's the idea of using physical chemistry as a tool, a lens to look at proteins. So now the dehydron concept has been used to understand disease, to do drug design, and to understand fundamental protein biophysics. Okay, but let's move on here. Now we're in the AI world, and as you probably know, AlphaFold has revolutionized the folding of proteins and also gotten a Nobel Prize. So what is that? How does it work? Well, what it does is it utilizes protein databases to predict structure. When you see a sequence, you say, how does this match up with what's in the database? And based on that, you say, okay, I predict that structure. But what Janet Thornton points out is that looking at ligand-induced protein folding is still a challenge today. And by ligand-induced protein folding, think of that picture that we had where a ligand was binding to a dehydron. That might be a situation where, in fact, upon that binding, the protein structure changes substantially. And she gives examples in this paper of that. So what we're going to do is deal with that fact that we want to be able to do protein folding accurately in situations where we don't have enough data to predict the fold. And so we're proposing a hybrid physics-informed AI approach. So the physics in question is the dehydron. We want to use the concept of dehydron to say, where could it be that we need to do something to fix up the AI AlphaFold predictions? And we're associating it with what we're calling structural plasticity. So this may not be a new term, but at least it was new to us, to identify this twilight zone Ariel talks about between order and disorder as being associated with structural plasticity. So what do we mean by that? Well, so structural elasticity is where the three-dimensional structure is very strong and it just vibrates around a little bit. Plasticity means that it can really be stretched. And so that's what we're saying these areas where dehydrons are present are likely to be. Okay, so like every machine learning tool, AlphaFold is limited by the available structural database for learning. Of course, this is not something that's generally understood by people on the street, that they think there's data everywhere, which it turns out not to be the case. And so they just think AI can solve every problem, which is certainly not the case. So many structures in drug target associations are underrepresented in structural databases. And it's because the ligand associations also often involve some structural deficiencies in the target protein. And it can be mitigated by a physics-informed AI to populate poorly represented regions of data space, fill in the data desert. And this is a paper that we published last year explaining all of this. And so I'm just going to quickly go through this paper and give you some highlights of that and then see if you've got some questions. Okay, so structural plasticity, it means proteins have some conformational diversity, but not fully unstructured. That is, they don't just flop around completely. That's the unstructured part. They're underrepresented in repositories of structural data just because they're not fully structured, and so they don't show up. And the dehydron model helps to identify such plasticity because we believe that this plasticity is often associated with dehydrons, because after all, dehydrons are weak hydrogen bonds, and that's what plasticity needs. Okay, and dehydrons are naturally underrepresented in structural databases since they lie at the tails of the strength and stability distributions of hydrogen bonds. And the biophysics concept of dehydron provides a way to compensate for the lack of diversity in existing protein databases, as we describe here. Okay, so plasticity does not mean disorder. However, Pondor, which does predict disorder, can be used to predict plasticity to some extent, because these transition regions from order to disorder, the structural twilight zones, the plastic regions, we believe all correlate together. So we think that something like Pondor can be used to tell us where do we look to see where there might be structural issues in existing proteins. Okay, so this is very common. Most signaling proteins have regions of plasticity that makes their interactions ephemeral and weak, and hence difficult to target, but that's how signaling works. And according to Janet Thornton, almost 2/3 of enzymes show conformational changes on binding their ligands. I was quite surprised about that, but I trust Janet Thornton. She's one of the leading lights in the field. So here what we're going to do is suggest how you could use Pondor and AlphaFold together in a pipeline to fill in the gaps. Okay, so let's look at Janet's data. Here are the two structures that she put into the protein data bank. One SW5 is the one on the right, and it's the one on the left, and one SW1 is the one on the right. So one is bound and one is unbound. And so the unbound state is on the left. And the main thing, there's nothing to learn from this except that they're different. And to some extent that there are more well-wrapped hydrogen bonds over here in, let me just make sure I'm saying the correct one. Yeah, so the left is the unbound state, and this is the bound state. Okay. So what happens is, upon binding, Janet Thornton did the experiments, you form a new structure here. Now, it's not a strong structure. It's got 3 dehydrons in this part of the sequence. First of all, you have to read carefully, but this is A205 glue, A205 glue here. This is just this backbone moving around the backbone to the other side, 213. So in the unbound case, AlphaFold gets the right structure, partly because it's got these well-wrapped hydrogen bonds that hold that part of the sequence together. But when you have it bound to a ligand, Janet Thornton found that it changes structure. And AlphaFold doesn't give this in any one of its top possible structures. It's quite different. And so there's something about this that needs to be understood, because often what at least Janet is saying is this is the picture you've got. Yeah, you know the structure, but what you don't know is what sort of structure you would get in one part of it. First of all, you don't even know to focus on this part. So what could you do to elicit information about possible, I'm going to say plastic regions where there are possible simple changes due to binding. And so what we do is we say, well, here's what we do. AlphaFold is getting the bulk of the structure correct, but the bound state differs substantially from the prediction. It does not show up in AlphaFold's top predictions. Not surprisingly, there are dehydrons there. And so what we can do is take a look at the disorder score. So that's a signal for plasticity. So suppose we're back up and we say, okay, we've come into Janet's lab. She's done the test, but she's not going to tell us what the answer is. She says, look, where should I look for binding? Well, we would say, well, we expect it to be in a plastic region. So we take Ponder and we run her sequence through it. And we find that there are certain dehydrons in certain places. And we say, well, maybe in these regions, we need to think about it. So let's just look at this picture here. So we take Ponder, we run the sequence through, and we find a particular transition zone right here that happens to be the region where we know the binding occurs. Of course, there are many other places you would have to look here, but they're finite in number. And we look in there and we say, okay, this is where we expect there to be dehydrons. And of course, there are dehydrons in there. And so we're saying that you can use these transition zones to predict the plasticity and then therefore get an idea of where binding could occur. Okay. Now, so that's our process. We use AlphaFold, homology modeling. We predict the twilight zones based on sequence. And then we're left with a small number of small plastic deformation regions. And so what we're going to do is we're going to simply say, well, since these extra regions are small, why don't we do some sort of brute force optimization? Okay, so that's the idea. So what we're doing is we're separating things into elastic, plastic, and disordered regions. We're focusing on the plastic regions. We know that actually Ponder and AlphaFold get the elastic parts exactly right. These are the ones where they differ, and these disordered ones are ones just where proteins don't do anything. Okay. And so we do local optimization. And so there are various techniques that have been proposed to do optimization of small proteins. And one of the most successful early ones was by Ariel Fernandez and collaborators at Chicago. Notice I am not on that list at that point. I was not offended by any means. They had plenty of co-authors. And they were folding what's called the 36 residue villain headpiece. So it's a 36 residue protein, and they did it by brute force, essentially. They use what are called Ramachandran basins to predict the structure. So that's a particular technique that can simplify things. And so they were able, I mean, it was not a short simulation, but by today's standards, that would be a relatively short simulation. And it's a pretty long sequence. This would be a very large plastic region. And so based on that, I'm saying this is feasible. Doing this sort of full-blown simulation of the structures is doable. And I point this out in part because Carl Fried and Tobin Sosnick were mentioned as John Jumper's advisors at UChicago. So we're very proud of all of this. And that's what we're proposing. Now, there are some simplifications one can make. So there's a backbone hydrogen bond network that any protein has. And so what you can do is you can write down the residue numbers here and here. And for each different structure, you can give us the hydrogen bond network. So this is the hydrogen bond matrix. It's a symmetric matrix. It's just an indicator matrix where there are hydrogen bonds. And in this version, we haven't said whether they're dehydrons or not. The idea being this, what you can do is you can say, okay, well, what I want to do is I want to predict a hydrogen bond network and then see if that's plausible or not. And so what you can do for any region of protein space, you can form such a matrix and just check to see whether or not you get that sort of interaction and use that as a way to test to see if you've got a fold of interest or not. So there's quite a bit you can do to reduce the hydrogen bond network matrices, and so I'll leave that for more reading in depth. And if you use Ramachandran basins, then that makes it all very computationally manageable. And then, of course, what you can do when you're putting all these supposed structures together, you get to put them back into the original AlphaFold structure and see if there are any clashes or not. If there are clashes, you have to throw it away and look for another one. Okay, so let me stop here and see if there are questions. I just want to say this has been based on joint work with mostly Ariel Fernandez, the co-author of our book, and Chris Fraser, who was a postdoc for many, many years. Harold Shiraga was very much involved early on in the study of wrapping and dehydrons. Christina Rogali was a student at Chicago, and while at Chicago, she worked with us before moving on to Princeton. And then Steve Beria and Tatiana Orlova at Chicago. And we were supported very generously by the Biophysical Dynamics Institute at the University of Chicago and NSF grants. And Ariel and I were fortunate to be able to be at the Mathematical Biology Institute at Ohio State during the fall of 2005. And then I'll just leave you with some references, and thank you very much for your attention.
Thank you very much, Ridgway. Let us thank Ridgway for the very nice presentation, please. Yeah.
Thank you.
Yeah, physically or virtually. Let me stop the recording.